So what I'm going to do is talk about active nematics. And the first part of the talk is really an introduction to active nematics and active turbulence. And what I want to try and get over is the idea of nematic symmetry and topological defects, active topological defects, and something called active anchoring. And then there's going to be another bit of sort of tutorial-like stuff, but it's something where I don't know the answer. I'm going to talk a little bit about epithelial cell layers, and probably people in this audience know more about it than I do. So I'll try and lay out the questions and what I think we don't understand, but it might well be just what I don't understand about how epithelial cell layers move. And then I'll do some talk about some recent results. And in particular, I thought I'd talk about cell sorting and some very recent work we've done on the distribution of tumors in breast cancer. And again, I really like to talk to people with a biological background, because do ask questions, because I'll learn a lot. So active matter. Active matter is meant to be out of thermodynamic equilibrium, and that's what's special about it. And because of that, it's interesting in many different contexts. And in particular, I'm going to talk today about biological systems, because I think that's one of the directions of active matter, to try and help understand developmental biology, the physicsy bits of developmental biology and of mechanobiology. So let's start with active nematics. These are the sort of systems I'm going to talk about. Dense active nematics. So a dense suspension of bacteria, microtubules, long, thin polymers driven by motor proteins, or a dense array of cells. And what you can see in all these pictures is there is chaotic motion, chaotic velocity fields. And that's a bit odd, because we're at low Reynolds number, and at low Reynolds number, you really aren't meant to get turbulent light flows. So what's going on? So this is the story of active nematics. So nematic symmetry is symmetry where you have a preferred axis, but the two directions of the axis are not distinguished. So there's no arrowhead on these molecules or colloids. And if you have loads of these nematic molecules or colloids together, they can form a nematic phase where they all tend to point in the same direction. And so you have long-range orientational order, but no long-range positional order. The center of these colloids don't have any order to them, but they all tend to point in the same direction. And there's an order parameter describing that. Sometimes it's described by the director field, which is just a headless vector in this direction. But what seems to work better is to use a Q-tensor. This Q-tensor is again an order parameter, and it tells you both about the direction of the ordering, the up-down direction, and also about the degree of ordering, how ordered these things are. So these things are very ordered, but often you can have a little bit of nematic order. So there's just a slight tendency to a preferential direction. And these nematics are fluids. So if you get a small bug in the order, if there's a fluctuation or something, and the nematic order gets changed like this, then that's fine. It's a localized distortion, and there are elastic forces in this system, so it's very easy for that to relax back and restore the order. However, you can have more important defects, which are called topological defects. And what I've done here is draw a picture of the most important topological defects in 2 dimensions. This one is labeled a +1/2 defect, and its shape looks like a comet. This one is labeled a -1/2 defect, and you get this 3-fold symmetry. And they're called topological defects because they muck up the order all the way out to infinity in an infinite system. You can't undo, if you like, this defect. It's a bit like a knot. And it has on its own an infinite energy because it distorts the order everywhere. So in fact, in an infinite system, you can't have a single plus a half defect. You have to have a plus a half defect and a minus a half defect appearing in pairs. And what they will do in just an ordinary non-active system, a passive system, is that they move towards each other very slowly and eventually either get stuck on some sort of dirt type defect or they annihilate each other. Now I'm going to say a little bit about the equations of motion. So if anybody isn't so used to the maths, don't panic. They're only about 2 or 3 slides like this, and it doesn't really matter if you're not used to it. What I really want to do is say, this is how we write down the equation of motion for passive liquid crystals, for non-active ones. How do we change that if we want to make them active? So these are equations a bit like the Navier-Stokes equations, continuum equations, but they're a little bit more complicated because I don't just have a simple fluid, I have a fluid which is made up of these long nematic particles. So first of all, I need an equation for Q, the order parameter. The order parameter is vectored by the flow. And then because these are long, thin molecules, it's turned by flow gradients. And then if I turn off the flow, whatever's making the flow, it has to relax to equilibrium. And equilibrium is described, as usual, by minimizing a free energy. And we take that as an expansion in the order parameter Q, the sort of bog standard Landau-de Gennes free energy. And then this is the elastic term. This says that if I distort these nematics in any way, I'm going to get an extra energy associated with them. So then I also need an equation for the velocity field. And the way I've got it written here, it looks just like the Navier-Stokes equation. The things on this side are the inertial terms, which you can sort of forget about at these tiny length scales. And then I have here just the derivative of the stress tensor, which is included in the Stokes equation. And that stress tensor has the normal viscous stress that you'd have in a simple liquid. And then it also has a thing called the elastic stress or the backflow stress. And what that is that if I've got these molecules and I'm moving them like that, they're going to set up a flow in the fluid. So that's well understood. Everyone sort of understands these are pretty nasty equations, but people believe them and they know how to solve them. So how do we make them active? How do we extend these equations to an active nematic? What's the extra physics we have to put in if this system is active? So what are we trying to describe? Well, we're trying to describe things which actually move around, which are self-propelled. And so they're moving around in a fluid. And what they're doing is they're setting up flow fields in this fluid. It's like if you're swimming in the swimming pool, you sort of set up flow fields around you, you make the swimming pool move. And the flow fields look like this around a single swimmer. These are the far flow fields. And you can see they have a very special symmetry. It's nematic symmetry, right? You've got this special axis, but this direction and that direction are the same if you're considering the far flow fields. This is real experiments from Ray Goldstein's group, very famous experiments. So that's the extra thing we have to put in. These extra flows, which have nematic symmetry, will give you an extra stress and extra forces in the fluid. So how does that appear in the equations of motion? Well, the answer is you just have this very simple extra term. It's an extra term which goes in the stress tensor. And this thing here is the activity that basically tells you how many swimmers you've got and therefore how strong these active forces are. And then it's proportional to Q, this order parameter. And that's because the flow fields have nematic symmetry. It's not entirely obvious why it's like that. That would sort of be another lecture. But it certainly makes sense that because of the symmetry of this flow field, you end up with a Q here, at least to leading order. So this comes in there. It comes in the stress. It comes under a derivative. So what it means is that every time you have a gradient in Q, or in zeta for that matter, but a gradient in Q will give you a non-zero term here, which means you have additional stresses and additional forces, and those are going to drive flows. If you had a load of swimmers which was perfectly ordered, you wouldn't get any active flows because you wouldn't have any gradients, but that soon becomes unstable. So really the punchline is that with this active term, gradients in the magnitude or the direction of the nematic order are going to induce stresses and lead to flows. Just a couple of words I'm going to use later on. These flows can either push outwards along the long axis of the active nematogen, swimmer if you like. That's called extensile or a pusher in swimmer language, and it corresponds to this zeta thing being positive. Or you can just have the flows going in the opposite direction. That's what's called a contractile active particle or a puller, and that corresponds to zeta is negative in these continuum equations. And it actually matters more than I expected. So now let's go back and say, what difference does it make? Okay, so we had the passive system, and a localized distortion would just relax to equilibrium very quickly. But now this is a gradient, right? I've got the nematic order is changing. And so I'm going to get stresses and forces and flows. And if you work out which direction they're in, then in this direction. And so that distortion is just going to get bigger and bigger. The system is unstable to a bend distortion. That's if it's extensile. If it's contractile, the flows are in the opposite direction and you're okay, but you get mucked up because if you have a splay distortion, one like that in the directive field, that's unstable. So these active systems are unstable to any sort of small fluctuation, and the nematic order is destroyed. And what you get instead is this state called active turbulence, this chaotic state. So this is a mixture of microtubules, long polymers, driven by motor proteins. And you can see they're doing this turbulent-like state called active turbulence. And, okay, it counts as a nematic. We might expect to see topological defects. And now the move is stopped. This is exactly what we see, right? So this one here is a very obvious +1/2 defect. And this one here with threefold symmetry is one of these -1/2 defects. So one gets topological defects in these systems. And whereas in a passive system, what happens is that the defects move towards each other and annihilate each other. What happens in an active system is not only can they be destroyed, but they can also be created in pairs of + and -1/2 defects. And they're created in pairs so that you end up with a sort of gas of topological defects, which are continually being created and destroyed, so that on average you have a well-defined average number of these defects. That's fine. The reason you can do it is the system is active. You're pumping in energy, and you are pumping an energy into the system, and that gives you the energy to create the defects. And what would happen in a passive system, occasionally you get a fluctuation which does create defects, but they'd immediately annihilate because they're next to each other. But in these active systems, this +1/2 defect is actually motile. It moves. And that makes sense, right? Because the directive field changes from place to place. There's a gradient in the directive field. Gradient in the directive field means that you have forces acting, and those forces push these defects towards their heads in an extensile system and towards their tails in a contractile system. These minus halves also produce flows because the director field is changing, but the flows balance because of the symmetry. They balance, so this thing is not self-propelled, but it is a source of flow fields. Right, so that's sort of the main conclusion on the active pneumatics. So just sort of two extra things there in terms of the background, which we'll think a bit about later, and because they're nice movies. So what happens if you take this active turbulence and you confine it? Well, if you put it in a channel, the active turbulence doesn't have room for the instabilities to get going, and you get laminar flow. Here's shear flow along the channel. This is microtubule motor protein mixtures, real experiments. You can see the shear flow, but then occasionally you get these topological defects forming. These are the plus or minus topological defects. So you get shear flow, and then occasionally you get these bursts of topological defects. We think that happens because the flow extends the microtubules, so they start looking nematic, but the nematic state has an instability, and so it undergoes its instability and creates these defects. These lovely, let's see if I can get this one to move, that's right. Same thing happens when you confine this to a circle. Again, you get the thing going round and round, the activity is making it go round, but occasionally you get one of these defects forming. And in a semi-regular way, which I think is not entirely understood, you get these defects being created at the surface, giving these lovely patterns. And of course, the same thing happens in in macroscopic systems as well. Okay, so the other thing I wanted to say was to tell you what active anchoring is, because that's going to be important for some of the research stuff later on. And the idea of active anchoring is that if you have an active system and you confine it, then there's an effective anchoring near the surface just caused by the activity. Nothing to do with thermodynamics, just the activity itself will anchor these things. So here I've got nematogens confined in a container. This is an extensile system, and you can see that they tend to align along the surface. In this one, which is a contractile system, they tend to align perpendicular to the surface. And the reason for that is active flows again, because at the surface, and here it's nematic, and outside it's not nematic, so the magnitude of the order decreases across the boundary. You have gradients in the order. Usual story, gradients in the order give you flows, and those flows are what is lining these things up. Just to show you the results of simulations, just to show this is really quite a strong effect. This is actually in 3D, ordering at the surface in an extensile system, where the light brown shows in-plane anchoring. This is in a contractile system, where the dark brown shows normal anchoring. And if I plot the angle between the director and, sorry, between the normal to the surface and the director field, you can see it really does want to be tangential in an extensile system, and it really wants to be normal in a contractile system. Okay, so let's stop there. Has anyone got any questions? Probably if you stick your hands up. No, please do. Yeah, okay. John, do you want to?
Yeah, sorry. A little earlier when you were showing the generation of flows by active, like bacteria and so forth, you showed a picture of what looked like a polar bacterium with a head and a tail, but it was producing something proportional to the quadrupole that had quadrupole order. Why wouldn't it be polar and have a dipole field?
I mean, the reason, no, it has a dipole field. What it doesn't have is a polar field, right? And it's because I'm looking at the far flow field. And that far flow field essentially is an average over time. If you look locally, obviously it is polar because it swims in one direction. And what tells you whether or not it swims is whether its stroke is the same forwards and backwards in time. But it's the far flow field we're worrying about here. And the reason the far flow field has to be dipolar, has to have basically flow like that, is that these are active particles, so you can't have a net force on them because there's nothing to balance the next net force. So Newton's law says I have to have a pair of forces like that, which gives me a dipolar far flow field.
Okay, thanks.
Okay, thanks for the question.
One quick question. Yeah. So in your examples, you had these long microtubule polymeric filaments, but also these sort of short nematic molecules, which is often how we think of liquid crystals. Does does length matter when we are thinking?
Yeah, that's a great question, and the answer is it seems no. It's going to have a small quantitative difference, but qualitatively, it really doesn't seem to affect the basic physics. So the continuum theory works both in both cases. The difference will be things like the size of the core of the defects.
Okay, thank you.
I mean, people worried about that, and it doesn't seem to be a big problem. Okay, so now I'm going to again talk about background stuff, but much less. I mean, I think what I've said up to now is accepted by everyone. What I'm going to say now is a lot of questions. And I'm going to ask whether epithelial cell layers can sensibly describe as active pneumatics. And I have loads of questions about this myself. So these are epithelial cells. They're a very common cell type in the body. They tend to line the cavities of the body, things like the cavities of the lungs, and the skin cells are epithelial cells. And luckily, they tend to like to be two-dimensional. And so we can use two-dimensional theories often, at least to start with, to describe what's going on. So I'm going to consider these cells when they're plated on a substrate. So they're taken out of the body, they're put on the substrate, and we might ask, how do they move? And we all understand how a single cell moves, that it sort of pushes on the surface to move with a polar force, but it's not so obvious how these things move. For example, what they can do is they can flock like this. They can all suddenly take off and move in the same direction. An example of this is that the cells inside the egg chamber of Drosophila rotate round and round. Or they can jam because they're stuck together, or if they're less dense, they do what these cells do here, which is move in a way which sort of resembles this active turbulence. I mean, now they're jamming, but when they started, they were moving around at random and doing active turbulence. I don't think anyone really knows which and why the cells choose one or the other mode of motion. For example, there's these old experiments now, but I still think they're really beautiful experiments. These are plated epithelial cells. And what the experimentalists from the Milan group did is that they looked at the velocity field. And here the velocity field is rather small and rather random and chaotic and looks like active turbulence. And then they added a chemical, Rab5A. And what happened is that the cells just took off and flocked with much larger velocities and very correlated velocities. So what I'm plotting here is the velocity as a function of time. This is a control where the Rab5A is not added. And this is the system where the Rab5A is added. And you can see that, they suddenly take off like mad, right? This is biological data, and you can actually tell a difference between the two data points. A huge difference. Let's look at the movies of the same things, right? I've played this a couple of times because it goes really fast. The one on the left is active turbulence, and the one on the right is the flocking. And you see there's a real big difference. So then you want to ask, you know, what's this RAB5A doing? And the answer you get from biologists is we don't really know, but it probably mucks up the cell junctions in some ways, the junctions between the cells in these layers. So nobody has ever been able to explain to me or anybody those experiments. What's going on in the underlying biology, which is causing this change in the physics, change in the motion. Let's just think a little bit more about active turbulence in these cell layers. Okay, because cells are round, yeah? I've been talking about nematic particles, which are long and thin. Why is it okay? Now, why am I calling this chaotic system active turbulence? Is that a fair thing to do? Well, here's some evidence that the answer is yes. All right, so these are pictures of, this one is human bronchial epithelial cells. And the picture at the bottom here is topological defects, where the red ones are -1/2 defects and the green ones are +1/2 defects. And you can see very nicely that you can identify topological defects in this system. Another bit of evidence that it's behaving like active matter is if you try and confine these cells. Here we've got active turbulence, but they're confined into a fairly small box. And what happens after a while is that they start going round and round, just like the confined systems that we saw. And okay, so why are these cells, which are round on average, managing to look like pneumatics? And the answer, I think, and there is theory behind this, although I'm not going to go into it, is because they're squishy cells, they can be extended. So here's a picture from our sort of original paper on this, which is that if you have a cell and a fluctuation can extend a cell locally. So one cell will go like this, but then because it's joined to its neighbors, it will also pull them to be extended. And so you'll get local nematic order because the cells are connected to each other. So what we've got here is a picture showing the local nematic orders just by looking at the cells in this little region and seeing its mean direction and so on. Here's the director field replotted so you can see what you're doing without the cells below. And what you can see is you do get short-range nematic order, which is what you need to get active turbulence. And you can look at topological defects, and this topological defect is happily moving towards its head. Okay, that's weird, right? Because cells are contractile. A cell will do this. So you certainly do get the defects, but they seem to be moving in the wrong direction. This is evidence that these things really are active defects. So what I'm plotting here is an experiment which Thuan Ben Suo carried out. Number of defects as a function of time. So what Thuan did is here he added to the cell layer, he added blebistatin, which is a sort of sleeping pill for cells. So he basically killed off the activity. So the number of defects decreased with time because there was no active motion to create defects. Here he washed out the blebistatin, which is like sort of waking these things up, and the number of defects increased again. So that's pretty strong evidence that this is an active effect. So then I talked about this, in fact, in front of Margaret Gardell, but several people, and they say, okay, but It's all very well. We believe these experiments, but my MDCK cells don't move. They really don't give active turbulence. So to explain that, well, not explain, but to show that that's sort of okay, there's some really nice recent work from Daniel Cohen's group at Princeton. Very simple experiment, but I think an important one. What he did was put this epithelial cell layer on the substrate as before, but here the substrate is coated with extracellular matrix, and here the substrate is coated with E-cadherin. So this would be like in a real system where the cells are in a biological system, the cells would be next to the extracellular matrix. And here is what you tend to put on slides to make the cells adhere and move around. Okay. And then they measured the mean speed as a function of time. And what you find is that on the ECM, these things do beautiful active turbulence with a large speed, which gradually decreases with time as they start to divide and jam. On this side, the things don't move. The velocity is very, very low. And this crossover is of order the width of one cell, actually less than the width of one cell. I mean, this that you can see here, the crossover is very, very sharp. So it obviously matters a lot where these cells are sitting on the material they're sitting on. And for some reason, they can sense it over very, very short length scales. So what's going on? Let's try and think about these cells in terms of simple active pneumatic theory. Let's start by asking what it gets right. Well, you do get chaotic flows, and there's quite a lot of evidence that they are like active turbulence. The velocity-velocity correlation functions agree between the simulations and what's happening in the cells. If you find the topological defects, if you measure the stress around the defects in experiments, it agrees very nicely with the theories. And these defects have been found all over the place now in biological systems. If you confine the cells, they move in a regular way. If you have, for example, a long, thin channel, there's some beautiful work by the Paris group, which shows that the cells will just move down the channel and matches beautifully with the active pneumatic theories. And in fact, circular cells are fine. As long as the circular cells can deform, you can extend the theory a bit to deformable active pneumatics, although we know how to start doing that, although we certainly haven't got to the end of it. And you can get this active turbulence. What didn't it get right? What didn't get right is nobody could really explain why topological defects move towards their heads, because that's what they're meant to do in an extensile system. But in a contractile system, they're meant to move towards their tails. Since the original paper, people have looked quite a lot at topological defects. And in fact, in many systems, they move in both directions. They move both towards their heads and towards their tails. And we think the reason for this is that the topological defects in whatever's producing the forces do move towards their tails, but the actual topological defects in the cell extensions can either be parallel or anti-parallel to those. You need two order parameters, so you need a different order parameter for the stress defects and the shape defects. So that's in the way to being sorted out. What is absolutely not being sorted out is the connections to the biology. I've talked about flocking, which presumably is due to polar forces. I've talked about active turbulence, which is produced presumably due to nematic or dipolar forces. What's producing those forces? The cells obviously different sorts of cells interact in a different way with the substrate. And you'd expect really probably a polar force on the substrate because the cells would be pulling on the substrate, trying to move in some direction. Also, the cells have junctions between them. And in the 2D models, these are just lines, but in reality, these cells have a height, and so they're areas. And they're definitely connected to each other through various adhesion junctions. And so the cells are pulling on each other, and those are going to be more like nematic forces. It seems you can switch from a nematic-like force to a polar-like force. You can switch from, in the experiments, the Milan experiments I showed you, these things flocked like a polar force, but they could also do active turbulence, and just adding a simple chemical seemed to make the difference. I don't think we understand where the forces come from in the models we're trying to write down. And in my view, that's the biggest question. And I think it's certainly something that people are arguing about, and I think perhaps they ought to argue about it a bit more. So I'll stop there in case anyone wants to tell me the answer to this, to how these forces are produced in biology, or to ask questions. And I'm sorry that's a bit vague. It's because I actually don't know the answer.